Then also students can book a slot for personalized counseling session so that they will have the direct interaction with the counselor. In Auricle, we have some subscription plan also starting from basic to premium. So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. So premium plan includes all the services that is provided by Auricle, like pre-recorded classes, live class, live doubt clarification, one-to-one personalized guidance, notes, quick revision, examination module. And the premium plan costs around 50,000. And also for instant 20% discount, you can use my code, that is Aanchal20. So, as we have started the internal exam marathon series, so there are some core features of this series, like rapid revisions of all the subjects, all the important topics, and also volatile facts, quick revision, and multiple choice questions will be covered, and also we assure top notes grading in the examination. So, let's discuss the important topics. So, in today's class, we're going to cover counter current mechanism, ADH, GFR, renal function test, cystometrogram, acidification of urine, diuretics, and renal clearance. So first we're going to proceed with JGA. JGA is juxtaglomerulus apparatus. Okay, so what is this JGA? JGA is basically the specialized organ that is situated near glomerulus of each nephron. Okay, and when we talk about the JGA, so JGA is formed by 3 different structures. So that we're going to discuss. The first we have macula densa. Then second we have extraglomerular mesangial cells. Then we have juxtaglomerular cells. Right. So as we can also locate these structures in the diagram also, we can see the macula densa is present here in between afferent and efferent arteriole. Then we have juxtaglomerular cells also here, right? And the location of this extraglomerular mesangial cells is here. Okay, so this is the location of extraglomerular mesangial cells. Okay, so now we're going to discuss about these particular cells and structures. So first we're going to start with macula densa. So macula densa is the end portion of thin ascending segment, as we can see, before it open into distal convoluted tubule. Okay, and as we can see in the diagram also, it is situated between efferent arteriole and afferent arteriole, right, of the same nephron. And also it is very close to afferent arteriole. And macula densa is formed by tightly packed cuboidal cells. As we can see, many cuboidal cells are there, right? So this is about the structure, that is macula densa. Now next we have extraglomerular mesangial cells. So these cells are situated in the triangular region. As we can see, this is the triangular region, right? So these are situated in triangular region bounded by afferent arteriole and efferent arteriole, and also by macula densa, right? So here macula densa is here, then efferent arteriole here, and finally afferent arteriole. So this is situated in the triangular region, okay? And also this type of cells also known as agranulal cells. And these cells secretes a very important hormone that is known as prostaglandins, and also they are responsible for the secretion of cytokines. Okay. Now finally we have this juxtaglomerular cells. Okay, so these juxtaglomerular cells, as we can see in the diagram, these are the specialized smooth cells. Okay, and they are situated in the wall of afferent arteriole, right? Like this is the afferent arteriole we have. So these cells are situated in the wall of the afferent arteriole. Okay, and these smooth muscle cells are mostly present in the tunica media and tunica adventitia of the wall of the afferent arteriole, and these cells are also known as granule cells, and because of the presence of secretory granules in their cytoplasm. Okay, now if we talk about the function of JGA. So the main function of the JGA to secrete very important two hormone. The first we have renin and second we have prostaglandin. Okay, so already about renin, we know that these cells are specialized to secrete renin, and renin is a peptide with 340 amino acids. I hope it's fine now, camera. Okay, so we have this renin with 340 amino acids, and along with angiotensinogens, the renin form the RAS system. Okay, RAS system is renin angiotensin system, and which is a hormone system that play a very important role in the maintenance of blood pressure. Okay, and also this prostaglandin, the prostaglandin is also responsible for the regulation of blood pressure. Okay, so this is all about the structure of the extraglomerular apparatus and the function. Now, next we have the another important one, that is the GFR. Okay, so the GFR stands for glomerular filtration rate. Stands for glomerular filtration rate. Okay, so what is GFR? GFR, the rate at which plasma is filtered by the glomeruli, and an important measurement in the evaluation of kidney function. And if we talk about the normal range of GFR, so it is 125 ml plasma per minute, or it will be 180 liter per day. Okay, and the plasma volume in 70 kg adult man is about 3 liter, and the kidney filtered the some plasma some 60 times in a day. Okay, so if we talk about the GFR, so GFR is a process by which the blood is filtered by passing through the glomerular capillaries by filtered membrane, and it is a first step, or it is the first process of urine formation, and the structures of filtration membrane is well suited for the process. Okay, so various factors are also there that is responsible for the regulation of GFR. That's also we're going to discuss. So factors we have: first, renal blood flow. Okay, so this is very important factor in the necessary of glomerular filtration, and it is directly proportional to blood flow. Okay, so if renal blood flow will increase, so ultimately the GFR will also increase. Okay, now next we have tubuloglomerular feedback. This one we're going to discuss just after this slide. Then third factor we have is glomerular capillary pressure. So this glomerular capillary pressure is again directly proportional to GFR. So if the pressure inside the capillary will increase, so ultimately GFR will also increase. Then another factor we have is colloidal osmotic pressure. Now, what is the colloidal osmotic pressure? Or colloidal osmotic pressure that is present in the solutes. Okay, so this is inversely proportional to GFR. Means if colloidal osmotic pressure will increase, so ultimately it will decrease the GFR. Okay, then next we have hydrostatic pressure in Bowman's capsule. So as the name suggests, hydrostatic pressure. Hydrostatic pressure means the pressure which is exerted by fluid, or which is exerted by water, right? So this hydrostatic pressure is again inversely proportional to GFR. Means if it will increase, then the GFR will decrease. Then another we have, if there will be constriction in the afferent arteriole. So obviously, if there will be constriction in the afferent arteriole, and afferent arteriole are those arteriole which are responsible for bringing blood to the nephron. If there will be constriction in the afferent arteriole, so it will be lead to low renal blood flow. So GFR will decrease. So it is also inverse. Then next we have constriction of efferent arteriole. Now, in constriction of efferent arteriole, we have two phases, or we can say we have two stages. The first we have initial stage, and second we have later stage. So what happened in the initial stage, as suppose this is the efferent arteriole, then this is we have glomerulus, then this is we have efferent arteriole, and this is we have Bowman's capsule. Okay, so suppose if the blood will come here from efferent arteriole, and then there is constriction which is found in efferent arteriole. So what happened? There will be backflow of blood towards the glomerulus. So ultimately at initial stage, what will happen? It will lead to increase in GFR. Okay, but if severe constriction are there, then what will happen? There will be more and more constriction, and there will be reverse backflow of blood, which leads to decrease in GFR. Okay, then along with this, other factors are also there, like sympathetic stimulation. So whenever sympathetic stimulation are there, severe sympathetic stimulation, so it will lead to decrease in GFR. And also if surface area of capillary membrane will increase, so more surface area, more filtration will be there. Okay, so more surface area, so more filtration and more GFR. So this surface area of capillary membrane will increase, then the GFR will also increase. Okay, so this is all about the factors which are affecting the rate of GFR. Now, next we have this tubuloglomerular feedback. Okay, so what happens in this case? This is the mechanism that regulates GFR through renal tubule and macula densa. Okay, and what happened? Macula densa of juxtaglomerular apparatus in the terminal portion of thick ascending limb is very much sensitive to sodium chloride in the tubular fluid. Okay, so when the glomerular filtrate passes through the terminal portion of thick ascending segment, the macula densa act like a sensor, and it detects the sodium chloride in the tubular fluid, and accordingly also responsible for its alteration. Okay, so what happens whenever there is increase in GFR? So it will lead to increase in NaCl concentration also. Okay, and if NaCl concentration will be there, then macula densa, the structures just now we have read, will get stimulated, and they will start secreting a very important enzyme known as adenosine, and they will cause the constriction of afferent arteriole, and finally it will lead to decrease in blood flow. Okay, so this is all about tubuloglomerular feedback. Now, next we have counter current mechanism, and counter current mechanism is a very, very important concept, and usually the questions asked from this topic. Okay, so when we talk about counter current mechanism, as we can see, this is a U-shaped tubule, right? U-shaped tubule, or we can say U-shaped tubes, in which the flow of fluid always occur in the opposite direction. It will be in reverse direction. Okay, so in two limbs of the U-shaped tubule, so we have two division here. We have vasa recta and we have Henle's loop. Okay, so vasa recta is basically the exchanger, and this Henle's loop is multiplier. Okay, so here if we talk about the Henle's loop, So this function as counter current multiplier, and it is responsible for the development of hyperosmolarity of medullary interstitial fluid and medullary gradient. Okay, and if we talk about the mechanism that occur in this, so if we talk about the loop of Henle, so first we have this Bowman's capsule. Okay, so what happens here? The glomerular filtrate collect at the Bowman's capsule here, and it is also very isotonic to plasma. Okay, so why this is because it contains all the substances of plasma except plasma protein, because the plasma protein cannot be filtered from here. Okay, so it remains as it is. So here the osmolarity of the filtrate at Bowman's capsule is about 300 milliosmo per liter. Okay, so now after this Bowman's capsule, we proceed towards this PCT. Okay, so what is this PCT? When filtrate flows through this PCT, there is an active reabsorption of sodium and chloride. So sodium and chloride will get active reabsorption and followed by obligatory reabsorption of water. So osmolarity of fluid remain the same as in the case of Bowman's capsule. That will be again 300. Here it will also remain 300, and in proximal convoluted tubule, the fluid is isotonic to plasma, right? Now next we have this this descending segment, okay, after PCT. Now what will happen when the fluid passes from PCT into the thick descending segment? Water will get reabsorbed because this is permeable to water, and this ascending segment is impermeable to water. Okay, so water is absorbed from tubule into outer medullary interstitium by means of osmosis, and it is due to increased osmolarity in the medullary interstitium outside the thick descending tubule, and the osmolarity of fluid inside the segment is between 450 to 600 milliosmo per liter. Okay, then if we come to this ascending one, okay, so when this ascending segment of loop ascend upward through the medullary region, the osmolarity will decrease. As we can see here, we have 1200 and it will gradually decrease. Okay, so the osmolarity will decrease, and it is due to the concentration gradient. Sodium chloride diffuses out of the tubule. Okay, because this segment is impermeable to water, but it is permeable to electrolytes. Okay, so osmolarity will decrease up to 200, and the fluid in this segment is slightly hypertonic to plasma. Okay, then finally we have DCT and this collecting tubule. Okay, so while entering the DCT, osmolarity of fluid will become 200. Okay, and it is again isotonic to plasma, and in the presence of ADH, DCT and collecting tubule become permeable to water, resulting in water reabsorption, and finally the concentration of urine will occur, and it is also found that collecting duct in P cells are there. So P cells are responsible for ADH induced water reabsorption. ADH induced water reabsorption. Okay, so reabsorption of large quantity of water increases and the osmolarity of fluid gradually, and when urine leaves the collecting duct, its osmolarity reaches the maximum of 1200 milliosmo per liter, the urine become hypertonic to plasma. Okay, so this is all about the Henle's loop. Now next we have the exchanger. The exchanger means the vasa recta. Okay, so here the vasa recta is also function as counter current exchanger, and it is responsible for maintenance of medullary gradient, which is developed by counter current multiplier. Okay, so here also vasa recta act like counter current exchanger because of its position, and again we can see in the diagram it is of U shaped, right, and with a descending limb and a ascending limb, and here we have this hairpin band. Okay, so what happened? The sodium chloride reabsorbed from the ascending limb of loop of Henle entered the medullary interstitium. It will be here, and from here it enters the descending one. Okay, so here from ascending to here it will enter the descending one. Okay, and simultaneously water diffuses from the descending to ascending. So water from here will move to this side, and the NaCl will move from this to this. Okay, so that the exchange between ascending and descending limb of vasa recta can be done properly. Okay, so this is all about the countercurrent mechanism. Now next we have the acidification of urine. So what is acidification of urine? So if we look at the picture also, the acidic means more number of hydrogen concentration and alkaline means less number of hydrogen ion concentration. And normally if we look at the normal body pH, so it is about 7.4. Okay, so metabolic, this is the metabolic function of body and are highly sensitive to pH. And if we look at the compatible pH for life, so it's range between 6.8 to 7.8. And acid-base balance is the coordinated function in lungs and kidney. So lungs are the major excretory route of volatile acid, that is carbonic acid. And kidney pH of urine range between 4.5 to 8, depend on the rate of acid secretion. So whenever body pH changes, the kidney start excreting acidic or alkaline urine and maintain acid-base balance of extracellular fluid. So in another word, we can also say that kidney plays a very important role in maintenance of acid-base balance by excreting more and more number of hydrogen ion and retain bicarbonate. Okay, so usually the kidney do excretion of hydrogen ion and retention of bicarbonate. Okay, so normally urine is acidic in nature and its pH range between 4.5 to 6, and metabolic activities are there in the body that produce large quantity of acid which threaten to push the body towards acidosis, and the kidney prevents this by doing two mechanisms, by reabsorption of bicarbonate and secretion of hydrogen ion. Okay. Then another thing is also there, that if we talk about the reabsorption of bicarbonate ion, so in kidney about 4320 milli equivalents of bicarbonate is filtered by glomeruli every day, and that is known as the filtered load. Okay, and excretion of this much bicarbonate in urine will affect the acid-base balance. Okay, so bicarbonate must be taken back from the renal tubule by reabsorption. Okay, then if we talk about the secretion of hydrogen ion, so reabsorption of filtered bicarbonate occurs by the secretion of hydrogen in the renal tubule. So about 4380 milli equivalent of hydrogen ion appear every day in the renal tubule by means of filtration and secretion. Not all the hydrogen ion excreted in urine out of 4380, about only 4280 to 4330 of hydrogen is utilized by the reabsorption. Okay, and only the remaining 50 to 100 milliequivalents is excreted. Okay, so this result in acidification of urine. Okay. Now next we have renal function test. So whenever we talk about renal function test, so first we should look upon the properties of urine. Okay, so when we look upon the properties of urine, so volume is around 1 to 1.5 liter per day. Then reaction is usually slightly acidic. Why it is slightly acidic? Because no number of hydrogen ions are there. Then specific gravity. So specific gravity is around 1.010 to 1.025. Then another thing we have osmolarity. So that is 1200 to 1500 milliosmo per liter. Okay, and the color is straw color. Okay, then if we talk about the renal function test, so in renal function test we do the examination. Okay, so in examination we have In examination, we have physical examination, microscopic examination, and chemical analysis. Okay, so one by one, we're going to know about the various examination. Okay, so if we talk about the physical examination, so in physical examination, we look for the volume. Okay, so whenever there is increase in urine volume, that indicates increase in protein catabolism. And some renal disorders are could be there, like chronic renal failure, diabetes insipidus, glucosuria. Okay, and then second we look for the color. Okay, so normally the urine color is straw colored, right? So it become turbid in both physiological and pathological changes. Okay, so physiological changes could be like causing turbidity of urine or precipitation of crystals, presence of mucus or vaginal discharge. And pathological condition that causes turbidity are presence of blood cells, bacteria, yeast. Okay. Then third, we have specific gravity. Okay, so specific gravity of urine is the measure of dissolved solutes or dissolved particles present in urine, and it is very low in the case of diabetes insipidus and high in case of diabetes mellitus. Then we have osmolarity. Okay, so osmolarity of urine that get decreases in case of diabetes insipidus. Okay, so this is all about the physical examination. Then also in physical, after physical, we do microscopic examination. Okay, so in microscopic, we do look for the presence of RBC, WBC, epithelial cells, or we look for the cast, presence of cast or the crystals. Okay, so presence of RBC in urine indicate the presence of glomerular disease, like whenever there is inflammation in glomerulus. Then presence of WBC that appear in high power field means whenever there is more number of WBC, it indicates glomerular nephritis or any kind of infection that is present in vagina, cervix, urinary tract. Okay, then epithelial cells. So normally few tubular epithelial cells are there that flow into urine, and presence of many epithelial suggests the presence of the condition like nephrotic syndrome and tubular necrosis. The next we have casts. So casts are basically the cylindrical bodies that are casted in the shape of renal tubule. So cast may be haline, granular, or cellular in nature, and haline and granular casts, which are formed by precipitation of protein, may appear in urine in small number. Then finally, we have crystals. So several types of crystals are present in normal urine, and common crystals are the crystals of calcium oxalate, calcium phosphate, uric acid, triple phosphate. Okay, so normally crystals are appear in the disease like liver disease. Okay, now next we have the chemical examination. So in chemical examination, we look for the glucose, then protein, then ketone bodies, bilirubin, bile salts. Okay. So glucose appearance in urine when the glucose blood level increases above 180 mg per deciliter. Protein presence of excess protein indicates the condition of proteinuria, and particularly albumin are majorly there. Okay. Then ketonuria means absence of presence of ketone body in urine that occurs in case of pregnancy, fever, diabetes mellitus, prolonged starvation. Then bilirubin is also there. That condition is known as bilirubinemia, and that occurs during the condition like hepatic or post-hepatic jaundice. Then finally we have bile salts, and bile salts, presence of bile salts in urine also reveal the condition of jaundice. Okay. One more thing is here, that appearance of blood. An appearance of blood in urine indicates hematuria. Hematuria means if there is any kind of renal stone, infection, or malignancy of urinary tract is present. Okay. Then other than these examination, we also do examination of blood. So for examination of blood, we look for estimation of plasma protein. Then we look for urea, uric acid, creatinine. Okay, so in these cases, we compare the normal range or in normal range of these urea, uric acid, and creatinine, and also we compare these normal range with the output that it has come. Okay, now next we have cystometrogram. Okay, so what is cystometrogram? Cystometrogram is the technique, okay, used to study the relationship between intravesical pressure and volume of urine in bladder. Okay, so basically this technique can be understood by the graphical presentation, as we can see in the diagram also. So what happened in this case, there is a method of recording this cystogram. So a double human catheter is introduced into the urinary tract of the patient. Okay. Now, what will happen? One human is used to infuse fluid into the bladder, and other one will be considered to use the record the pressure changes by connecting it to a suitable recording instrument. Okay. Now, what we will do, the bladder will get emptied completely, and then a known quantity of fluid will be introduced into the bladder at regular intervals. Okay, that intervals also we're going to discuss. Then the intravesical pressure developed by the fluid is recorded continuously, and a graph will be obtained. Okay, so how the graph will be there? Like in graph, as we can see, we divide it into 3 segments. Okay, so first we have first segment, then second segment, and finally the third segment. Okay, so what is there in first segment? So initially when the urinary bladder is empty, the vesical pressure is 0. Okay, so here we have this pressure is 0, and when about 100 ml, so here we can see 100 ml of fluid is collected, the pressure will rises up to this 10. Okay, 10 centimeter per H2. Then as we can see in the diagram, this segment 2. So segment 2 is showing the plateau curve. Okay, plateau, or we can say no change in the intravesical pressure, and it remains at 10 centimeter H2O even after introducing the volume up to 400. We can say as no such changes are there. Okay, and why this changes are not occurring? It is because of the adaptation of urinary bladder by relaxation. Okay, and this is also considered in the law, that is P is equals to T upon R. Okay, so according to law, this is law of Laplace. So according to law, the pressure in the spherical organ is inversely proportional to radius. Okay, so the tone remaining will be constant here. Okay, then finally we have this third segment. Okay, so after 400 ml of fluid that get collected, there will be contraction of the detrusor muscles. So detrusor muscles are those muscles which are present in urinary bladder. So become very intense, and there will be increase in the consciousness and the urge of maturation, and still the voluntary control is possible up to 600 to 700 ml, at which the pressure will rises up to 35 to 40. Okay, so this is all about this cystometrogram. Then next we have the another one, that is diuretics. So what are diuretics? These are the agents, or we can call these are the substance which enhance the urine formation and output. Okay, so these substances increases the excretion of water, sodium, chloride through urine, and diuretic agents increases the urine formation